Hi everyone, welcome back to class. And this today's class will be the last class on our revision class about nutritional biochemistry. And today, what we want to talk about is foodborne diseases. Foodborne diseases. So that is what our revision will be based on. What are foodborne diseases? They are illnesses that are caused by consuming contaminated food substances. And this foodborne disease that we're talking about could be classified into two types. We have the foodborne infection and we have the foodborne intoxication. Let's be taking them one after the other. Foodborne infections are caused by pathogens, and these pathogens includes bacteria, they could be fungi, they could be viruses, etc. They are caused by pathogens, which are disease causing microorganisms. But for foodborne intoxication, they are caused by toxicants, and the toxicants could be biotoxins that is even gotten from pathogens. Then also it could be accidental addition, it could be intentional addition, it could be gotten from metabolic sources, etc. So that is the main difference between both of them. The one is caused by once contamination is caused by pathogen, the other one is caused by toxicants. Now, for our revision today, let's focus our attention on foodborne infection. And we said they are caused by pathogens. So we will try and look at like two to three infections, and we will describe and revise them based on six main classification. What I mean by that is we will take each infection and discuss them based on the pathogen that cause this infection, the mode of transmission of this infection, clinical symptoms of this infection, diagnosis of this infection, treatment of this infection, control measures of this infection. These are the six important aspects that we will look at or about each of these infections. Let's start. The first infection that we'll be discussing today is cholera. Now, the pathogen responsible for that infection is called cholera. Then, for how this pathogen work is that upon infection, that means upon entering of this pathogen into the system, then there is multiplication of them in the small intestine. Then, which further now leads to the release of enterotoxin. They release enterotoxin into the system. So this enterotoxin leads to the stimulation of persistent outpouring of isotonic fluids by the gut mucosa cells. So moving on, the mode of transmission of these infection. One, it could be transmitted through the fecal oral route. It could be transmitted through contaminated water. It could be transmitted through contaminated food. Then even man is a natural host of this pathogen, so it could be transmitted through improper hygiene also. Now let's move on and check clinical symptoms of this infection. One, it include watery diarrhea, then effortless vomiting, then it also include dehydration, then electrolyte imbalance. And when there is no fast action taken, it could lead to other complications such as low blood pressure, wrinkling of the skin, sucking of the eye, which will eventually lead to death. So, diagnosis of this infection. One, bacteriological examination will be done on the stool that was passed by the individual. Also, the food that the individual took will be tested. Then also the water also will be tested just to know if there is presence of this pathogen in them. Then treatment of this infection. One, fluid replacement. You know that the person has lost a lot of fluid before, so fluid replacement. Then antibiotics will also be administered to the person, but the most important thing is that the person will have to visit the nearest healthcare center to be treated appropriately. Now, the control measure for that infection include proper sanitation, proper hygiene, safe water treatment, then vaccination. Let's move on to the second example that I want to talk about today. That is typhoid fever, and it's also caused by bacteria. So typhoid fever. Let's start with that. Pathogens that cause this typhoid fever is Salmonella typhi. Then the mode of transmission of these pathogens, of these infections, include contaminated food, also contaminated water. It is often also transmitted through fecal oral routes, then also improper hygiene. Then symptoms of these infection include fever, it include headache, it include abdominal cramp, it include constipation, it include diarrhea, etc. Then diagnosis of this infection: blood tests will be carried out on the person infected. Also, the stool that the person has passed out will be tested to see if this pathogen is present. Then treatment of this infection: number one, antibiotics can be prescribed from the person, but as I always say, the person should make sure to visit the nearest healthcare center for proper treatment. Then control measure to prevent these infections include proper sanitation, proper hygiene, safe water treatment, and at times sef vaccination could work. Now, moving on, we've discussed two examples under foodborne infections that are caused by bacteria. Now let's check the ones that is caused by virus. Under this, under this, the example we'll be taking is hepatitis A. Hepatitis A. Now, for pathogens that are responsible for hepatitis A is hepatitis A virus. That is the pathogens that cause this infection. Then the mode of transmission of this infection include through contaminated water, through contaminated food, um through fecal oral routes, through coming in contact with person that is already infected with this pathogen. And the incubation period for this virus is between 14 to 28 days, meaning that somebody can be infected today and will not see symptoms of that infection until the next 28 days. And that's the main difference between foodborne infection and foodborne intoxication. Foodborne infection always have longer incubation period, but foodborne intoxication has shorter incubation period. You could be seeing symptoms within minutes, within hours. In some days you will see symptoms. But for foodborne infection, it could take days, weeks before symptoms are being shown. So the symptoms now include fatigue, loss of appetite, nausea and vomiting, abdominal pain, then dark urine and pale stools, then also jaundice, which is the yellowing of the eyeball. And you know that is also telling us that something is actually wrong with the liver. Then the diagnosis of this infection is also one, blood test. Then also we check the stool that the person has passed, if this pathogen is actually present there. Then the next one is the treatment. As I always say, the person should visit the nearest healthcare center for proper checkup. But in addition to that, the person should also rest a lot, then stay hydrated, and also the person should be eating balanced diet. It will also help. Then preventive measure Control measure for this infection include proper sanitation, proper hygiene, safe water treatment, good personal hygiene, then also vaccination. With this, we've come to the end of our revision series on nutritional biochemistry. I hope this has been a big help to you, and I wish you all the best in your exams. See you in the next class. Bye bye.